Histones are loosely packed chromatin, densely packed chromatin, positively charged basic protein. So A is 3. A3, A3 is here, and option 3 no, and in option 4. So B is either 1 or 4. B is nucleosome. 1 is loosely packed chromatin, no. So it will be 4. B will be 4. DNA wrapped around histone octamer. So A3B4 is in option number 4. So strike this thing out. Right answer is option number 4. Yes. So we did it in less than 45 seconds. Your approach has to be QCQ. Quickly, correctly, and quietly. And check how much time are you taking in solving match questions. The lesser the time, the better it will be. So let us understand every point asked in this question. Histones they are structures, ball-like structures made up of proteins which are rich in lysine and arginine. This is given in the textbook, so make a note of it. These two are positively charged amino acid, positive amino acids. Okay, so positive charge on the histone protein helps in holding the DNA molecule around it. So we can nicely wrap the DNA around the histone octamer. So DNA is negatively charged because of the phosphate groups. Okay, so thermodynamically it is easy to wrap DNA around this. You don't have to supply extra ATP for the histones to hold DNA. The electrostatic force of attraction helps in wrapping DNA around it. And this is the way DNA is nicely organized in the nucleus. So we are clear with this and this. Next aspect which is asked in the question is nucleosome. Nucleosome is the structure which is formed by histone protein. The details of histone protein is not given. If you come across any other reference book, they will say histone is made up of these proteins. So pairs of these 4 proteins form the octamer. Okay, and then there is another protein which is called H1, that is also histone protein. So together the DNA wrapped around this octamer plus little bit of DNA on this, the DNA which is present on H1 is called linker DNA. Okay, linker DNA. So the total amount of DNA is 200 base pairs. So you might get mathematical questions based on this information. So one nucleosome has 200 base pairs. So maybe they will ask you how many nucleosomes are present if the DNA size is 2000 base pairs. Okay, so you need to understand how to do the math. This is simple cross multiplication. One nucleosome has 200 base pairs, so 10 nucleosome will have 2000 base pairs. So they can change the number and ask you, or vice versa. They can ask you what is the size of DNA if these many nucleosomes are present. So now the last segment, euchromatin and heterochromatin. Euchromatin and hetero. So these are also called, this is called the light region and this is the dark region, because upon staining this stains, this is darker upon staining and this is lighter, and the reason is this is loosely packed. So why this is loosely packed? Because it can be accessed regularly. So this is transcriptionally active, and the genes are expressed. Expressed. And the darkly stained regions of chromatin, they are tightly packed because they are not regularly accessed, and that is the reason they are known as transcriptionally inactive. And since they are transcriptionally inactive, these are not expressed. The genes are not expressed. So everything is connected. So they might ask you which segment of chromatin is not expressed, or they can ask you which segment of chromatin is tightly packed, or which segment is transcriptionally inactive, or the segment of chromatin which stains dark. So all these questions will have single answer, which is heterochromatin. One thing you need to understand is euchromatin and heterochromatin together forms the chromatin. The total DNA present in the cell. So the ones which are not expressed are not lost by the cell. They are still kept intact. And there's a reason behind it. If the cells are required for to regenerate other tissues, there is always a possibility of de-differentiation. So a set of transcription factors will help in again conferring the ability to divide on these cells. And this is the reason why the chromatin is not lost. The genes which are not used are not lost. They are stored inside the cell. Only thing, they will be tightly packed. And the ones which are regularly used are loosely packed, called as euchromatin. So euchromatin region stain light, and they are loosely packed, they are transcriptionally active, and these genes are expressed regularly. So this was all about all the topics which were asked in the question. I hope this video was helpful to you. Thank you so much for watching my videos.